Hello and welcome. Today we're going to be talking about bacteria and archaea. Hello and welcome. This is part one lecture covering the bacteria and archaea. Archaea. If you're kind of confused how to pronounce that word, just think IKEA and kind of change it a bit to sound like archaea. So we've previously talked about the organization of living things. We have prokaryota and eukaryota, okay, that are composed of prokaryotic cells or prokaryotes and eukaryotic cells or eukaryotes, okay? So those are the two main cell types, prokaryotes and eukaryotes. And so let's first talk about the eukaryotes, which is most everything that you see around you and can identify with. So we have got animals, plants, fungi, protists, and prokaryotes are the two other things we'll talk about there in a second. So we're very familiar with animals. Beetles, for example, or insects is the majority of animals on the planet of Earth, okay? You might think horses or, dogs or something, but actually beetles far outnumber, not beetles, but insects in general far outweigh or outnumber the other organisms that are in the plant kingdom. Cats, of course, are mammals. Frogs are amphibians. Lizards here are reptiles. And I'm just giving you a brief overview of the main kind of types of animals. Fish are, of course, aquatic living, and birds, for example. Okay. Okay. We've got plants, okay, which are autotrophic. This means that they obtain their own energy not by eating something else, but just basically by photosynthesis from the sunlight. So these are the main types of classes of plants. We've got ferns, we've got flowering plants, like these flowers here. We've got pine trees, and then here's an example of a moss. Okay, those are very typical types of plants there. We've got fungi, and fungi, it's not referring to a college student that's fun to go out with, but it's referring to, in many cases, we think mushrooms, for example, but there's many different classes, okay? So different types of molds, like this bread mold, are a type of fungi, and you can see how there's a budding kind of thing that can break off and produce spores in a zoom in of that. We've got sac fungi down on the bottom left, and there's basically a formation of a bulb that forms a sac and opens up in that way. And we've got, of course, the mushrooms, which are there in the middle. And then on the right, we have something called the imperfect fungi. It's kind of like weird, but it is a fungi, okay? So again, biologists put these into different classes depending on their shapes, their morphology, how they move, how they obtain nutrients, how they reproduce, and so on and so forth. Finally, we'll talk about Protista. These are unicellular organisms like amoeba, giardia, dinoflagellates, slime mold. Here in the middle, we have some algae. So red algae, green algae, many different kinds of algae are an example of a Protista. Up here on the upper right is slime mold. And this one here is affectionately referred to as dog vomit slime mold. Okay. So if you see that around the forest floor, that's what that is. Explore that at your own risk. Now, what are the main differences between prokaryotes? So remember, all the things we've talked about up to this point are eukaryotes, plants, animals, okay, fungi. Now we're going to be talking about prokaryotes, which are bacteria and archaea, okay? So the cell types are exactly the same, the cell morphology is exactly the same, but here we see a difference in the cell wall. We'll talk about this in the next class. The cell membrane type is exactly the same, and what's different is the plasma membrane. Okay, the lipids are slightly different that make up the different cell walls and lipid membranes. Okay, the chromosomes are both circular, they're the same, and then these bottom portions are different. Okay, so some of the cellular machinery is different within these two types of prokaryotes. Okay. So here's a picture of hot springs. Now, archaea are generally extremophiles. They live in crazy environments, very high pH or very high temperature, or they can survive under the Arctic ice at very low temperatures. So we're going to learn about some very interesting differences between typical bacteria and archaea. Okay, so and I'm going to talk a little bit about evolution and the history of the Earth. And again, it's a theory, it's not fact, and it should not contradict anything that you might hold in terms of your religious beliefs, okay? So, again, it's just a theory. It's not saying it's fact, but here we go. So in the recent past, scientists grouped living things into 5 kingdoms: animals, plants, fungi, protists, and then prokaryotes, okay, based on several criteria such as the absence or presence of a nucleus in the cell and maybe other membrane-bound organelles, the absence or presence of cell walls, multicellularity, and so on. Now, most recently, the pioneering work of Carl Weiss and others compared sequences of small subunit ribosomal RNA, which resulted in a more fundamental way to group organisms on Earth. So this again is the advances in RNA and DNA classification of these guys, okay? So the domain called bacteria comprises all organisms in the kingdom bacteria. The domain archaea comprises the rest of the prokaryotes. And the domain eukarya comprises all eukaryotes, including organisms in the kingdom Animalia, Plantae, Fungi, and Protista. Now, 2 of the 3 domains, bacteria and archaea, are prokaryotic. Now, prokaryotes were the first inhabitants on Earth, appearing 3.5 to 3.8 billion years ago. These organisms are abundant and ubiquitous. That means they're found everywhere, from bottom of the ocean to near volcanoes and on your skin and everywhere, okay? In addition to inhabiting moderate environments, they are found in extreme conditions, from boiling springs to permanently frozen environments in Antarctica, from salty environments like the Dead Sea to environments under tremendous pressure such as the depths of the ocean, and from areas completely lacking oxygen, such as a waste management plant, and even found in radioactively contaminated regions such as Chernobyl, okay, which, as you know, is in Ukraine if you've been watching the news. Prokaryotes reside in the human digestive system and on the skin, and they're responsible for certain illnesses and serve an important role in the preparation of many foods, okay? Our first kind of gallery picture here are some microbial mats or large biofilms, and they may represent the earliest forms of prokaryotic life on Earth. Now, there is fossil evidence of their presence starting about 3.5 billion years ago, and so it's rather remarkable that cellular life appeared on Earth about 1 billion years after the Earth itself formed, suggesting that pre-cellular life that could reproduce itself had evolved much earlier. And this is quite amazing if you think about it. A microbial mat is a multi-layered sheet of prokaryotes, okay, that includes mostly bacteria, but also archaeans. Microbial mats are only a few centimeters thick, and they typically grow where different types of materials interface, mostly on moist surfaces. The various types of prokaryotes that comprise them carry out different metabolic pathways, and that is the reason for the various colors that you might see here. And prokaryotes in a microbial mat are held together by a glue sticky substance that they secrete called extracellular matrix. The first microbial mats likely obtained their energy from chemicals found near hydrothermal vents. Now, hydrothermal vents are a breakage or a fissure in the Earth's surface that releases geothermally heated water. With the evolution of photosynthesis about 3 billion years ago, it is theorized that some prokaryotes in microbial mats came to use a more widely available energy source, which was sunlight, whereas others were still dependent on chemicals from hydrothermal vents for energy and food. Fossilized microbial mats represent the earliest record of life on Earth. A stromatolite, shown here on the left, is a sedimentary structure formed when minerals are precipitated out of water by prokaryotes in a microbial mat. Stromatolites form layered rocks made of carbonate or silicate, and although most stromatolites are artifacts from the past, there are places on Earth where stromatolites are still forming. For example, growing stromatolites have been found in the Anza-Borrego Desert State Park in San Diego County, California. And I believe this figure on the left is the microbial mats you can find in this park in San Diego, which is in very southwest California. And on the right are some rocks. Basically, the material from these stromatolites have fossilized over time, and that's not a rock. It's a fossil, okay? It's kind of like petrified wood. It's a living creature that was changed into a solid rock type of material. So we can do carbon dating and figure out how old these things were, and they are very, very old, okay? So again, this is just the theory of how life may be developed on the planet Earth. But so here's a person standing by, this little stream running off of a hydrothermal feature called a hot spring, I think, in Yellowstone National Park. And it's kind of greenish because of the cyanobacteria that it contains. So evidence indicates that during the first 2 billion years of Earth's existence, the atmosphere was anoxic. This means that there was no molecular oxygen, okay? Anoxic. Therefore, only those organisms that can grow without oxygen, we call these anaerobic organisms, were able to live. Now, autotrophic organisms that convert solar energy into chemical energy are called phototrophs, and they appeared within 1 billion years of the formation of Earth. Then, cyanobacteria, also known as blue-green algae, evolved from these simple phototrophs at least 1 billion years later. It was the ancestral cyanobacteria, again, that has been theorized, that began the oxygenation of the atmosphere. Increased atmospheric oxygen allowed the evolution of more efficient oxygen-utilizing catabolic pathways. It also opened up the land to increased colonization because some O2 is converted into O3, which is ozone, and then ozone, as we know, effectively absorbs the ultraviolet light that could have otherwise caused lethal mutations in DNA. And again, it's theorized that all this evidence suggests that the increase in O2 or oxygen concentrations allowed the evolution of other life forms. So again, it's theorized that Archaea or bacteria under the ocean is protected by the water layers from ultraviolet light. And since you don't need oxygen, it's fine. But once there were cyanobacteria that produced oxygen as a byproduct, then it opened the route for ozone formation, so things could live on the surface of the ocean or a surface of land, and also things didn't need to be right next to a water source. Things could maybe survive in the desert or a more dry area. Okay. So let's talk about extremophiles. As I mentioned, Archaea are extremophiles. Now, some organisms, and there's a table here that we can look at, some organisms have developed strategies that allow them to survive extremely harsh conditions. Almost all prokaryotes have a cell wall, a protective structure that we've talked about that allows them to survive in both different salt environments. Okay, some soil bacteria are able to form endospores that resist heat and drought. And so it allows the organism to survive unfavorable conditions until the weather returns to a good condition, and then these bacteria can, kind of grow again. Okay, these kind of adaptations, along with others, allow bacteria to remain the most abundant life form in all terrestrial and aquatic ecosystems. Prokaryotes thrive in a vast array of environments. Some grow in conditions that would seem very normal to us, room temperature, room pressure, moist, nice humidity, and so on, whereas others are able to thrive and grow under conditions that could kill a plant or you or me. Bacteria and archaea that are adapted to grow under extreme conditions are called extremophiles, which means lovers of extremes. Extremophiles have been found in all kinds of environments, the depths of the oceans, hot springs, the Arctic and the Antarctic, in very dry places, deep inside the earth, in harsh chemical environments, and in high radiation environments, just to mention a few. Because they have specialized adaptations that allow them to live in extreme conditions, many extremophiles cannot survive in moderate environments. So something that can survive in boiling water in a hot springs in Yellowstone cannot survive in your bottled water, which is at room temperature. So I find that amazing. Now, there are many different types of extremophiles, and they're identified based on the conditions in which they grow best. And several habitats are extreme in multiple different ways. For example, a soda lake is both salty and very basic or alkaline. So organisms that live in a soda lake must be both alkalophiles and halophiles. And there's a little table up here showing kind of that. Other extremophiles, like radio-resistant organisms, do not prefer an extreme environment. In this case, one with high levels of radiation, but they've adapted to survive it. I'll show you a figure of that. Organisms like these give us a better understanding of prokaryotic diversity and open up the possibility of finding new prokaryotic species that may lead to the discovery of new therapeutic drugs or have industrial applications. Now, this is a radiation-tolerant prokaryotes. Okay. Here it's a picture visualized in the false color transmission electron micrograph. It's a prokaryote that can tolerate very high doses of ionizing radiation. It has developed DNA repair mechanisms that allow it to reconstruct its chromosome, get this, even if it has been broken into hundreds of pieces by radiation or heat. This was discovered by somebody that took a bacterial soup, and they put it inside of a nuclear reactor, and they radiated it for like a week. And they took it out, and because of the high temperature and the high radiation, they figured, okay, all these bacteria are going to be obliterated to smithereens. They took the sample out, and then they found some surviving bacteria in there, and they were just amazed. Okay, again, these are extremophiles. Okay. Halophiles, here it is. One example of a very harsh environment is the Dead Sea. Okay, it's a hypersaline basin that's located between Jordan and Israel. Hypersaline environments are essentially concentrated seawater. In the Dead Sea, the sodium concentration is 10 times higher than that of seawater, and the water contains high levels of magnesium, about 40 times higher than seawater, that would make it toxic to most living things. And there's other stuff, iron, calcium, magnesium, that produce what is commonly referred to as hard water. Now, taken together, the high concentration of divalent cations, the acidic pH, and the intense solar radiation flux make the Dead Sea a unique and uniquely hostile ecosystem. Now, what kind of prokaryotes do we find in the Dead Sea? Well, the extremely salt-tolerant bacteria mats, including Halobacterium, I can't pronounce this other one, it's found in other locations, not just the Dead Sea, okay? And so on and so forth and so on, okay? There's many different types, but here's a little picture of one, okay? So these guys here can survive in extreme environments, okay? Now, the process of culturing bacteria is very complex and is one of the greatest discoveries of modern science. Now, there was a German physician by the name of Robert Koch, who is credited with discovering the techniques for pure culture, including staining and using growth media. Now, microbiologists typically grow prokaryotes in the laboratory using an appropriate culture medium containing all the nutrients needed by the target organism. The medium can be liquid, broth, or solid. Now, after an incubation time at the correct temperature, there should be evidence of microbial growth, as you can see here on the upper left. Koch's assistant, Julius Petri, who, by the way, invented the Petri dish, whose use persists in today's laboratories. Now, Koch worked primarily with Mycobacterium tuberculosis, that causes tuberculosis, and developed guidelines called Koch's postulates to identify the organisms responsible for specific diseases. Now, he was a physician, right? A doctor, so this is why he was doing this, all right? Now, his postulates include that an organism can be identified as the cause of disease when it is present in all infected samples and absent in all healthy samples, and it is able to reproduce the infection after being cultured multiple times. Today, cultures remain, of course, a primary diagnostic tool in medicine and other areas of molecular biology. Koch's postulates can be fully applied only to organisms that can be isolated and cultured. Some prokaryotes, however, cannot grow in a laboratory setting. In fact, over 99% of bacteria and archaea are unculturable. Let me repeat that. That's quite amazing. Over 99% of bacteria and archaea are unculturable. We put it on a little petri dish, we try to grow it, it does not grow. So we can't isolate the wonder drugs and anti-cancer medicines from those bacteria or archaea to develop them. Now, for the most part, this is due to a lack of knowledge as to what feed these organisms and how to grow them. They may have extremely special requirements for growth that just remain unknown to scientists, such as needing specific micronutrients, the pH, temperature, pressure, cofactors, or co-metabolites. Some bacteria, as we'll see, cannot be cultured because they're parasites. They need to live within a different cell, and they can't be grown outside of the host cell. All right, let's begin talking about bacteria. Now, there are many differences between prokaryote and eukaryote cells. The name prokaryote suggests that prokaryotes are defined by exclusion. They are not eukaryotes, or organisms whose cells contain a nucleus and other internal membrane-bound organelles. However, all cells have four common structures: the plasma membrane, which functions as a barrier for the cell and separates the cell from its environment; the cytoplasm, a complex solution of organic molecules and salts inside the cell; a double-stranded DNA genome, the information, of course, that's the blueprint for how that cell acts and reproduces; and ribosomes, where protein synthesis takes place. Now, prokaryotes come in various shapes, but many fall into three categories: cosi or spherical, there on the left; bacilli or rod-shaped, there in the middle; and spirally or spiral-shaped, here on the right. You can barely see kind of like this spring kind of looking thing on there. That's the spiral-shaped cell. Okay, so the cell is like this weird spiral shape. Now, recall that prokaryotes are unicellular organisms that lack membrane-bound organelles or other internal membrane-bound structures. Okay, you can see that in this figure here. Their chromosome, usually single, consists of a piece of circular double-stranded DNA located in an area of the cell called the nucleoid. Most prokaryotes have a cell wall outside the plasma membrane. The cell wall functions as a protective layer. It's in light green there, and it's responsible for the organism's shape. Some bacterial species have a capsule outside the cell wall that's in a lighter green, and the capsule enables the organism to attach to surfaces, protects it from dehydration and attack by phagocytic cells, and makes pathogens more resistant to our immune responses. Now, some species have flagella. The singular word for flagella is flagellum. And this is like a little tail-like thing, okay? And that's used for locomotion. And pili, okay, there's an arrow pointing to one of those, is a little hair-like feature, and that's used for attachment to surfaces, including the surfaces of other cells. Plasmids, which consist of extra chromosomal DNA, are also present in many species of bacteria and archaea. So recall, this is kind of like a good overview here. Recall that prokaryotes are divided into two different domains, okay? Bacteria, shown here in green, and archaea, shown here in blue, which together with eukarya comprise the three domains of life, okay? Now we'll talk about Protobacteria. The characteristics of bacterial phyla are described in these two figures. I'll zoom in on each of the parts in a bit. Major bacterial phyla include the Proteobacteria, the Chlamydias, the Spirochetes, the photosynthetic Cyanobacteria, and the Gram-positive bacteria. The Protobacteria are in turn subdivided into several classes from the alpha to Epsilonproteobacteria. So let's go through these galleries here and just introduce ourselves to the different things living around, okay? So here we have alpha Proteobacteria, and you can read along too here or pause the video. Now these are, an example of this is the Rhizobium, which is a nitrogen-fixing endosymbiont associated with the roots of legumes. So there's actually bacteria that live on the roots of, say, soybeans, for example, and they're symbiotic. Okay, the plants need the bacteria, and the bacteria need the plants. The bacteria help with nitrogen fixation and basically fertilize the soil so that the soybean can grow better, okay? There's also the good and the bad with many of these slides. So we have Rickettsia, which is a parasitic bacteria that causes typhus and something called Rocky Mountain spotted fever, okay? And there's a picture of what it might look like. And this is inside of a host cell, okay? So the bacteria is actually living within a cell. Here's the beta Proteobacteria. An example of this is the nitrosomas, which can oxidize ammonia to nitrite, okay? Here's a bad one. Spiralium minus, it looks like, causes rat bite fever. I don't think I want to Google what that is, but that does not sound good. And so here's a little picture of it under a microscope, and you can see how almost looks like a spiral-shaped bacteria there. Gamma Proteobacteria. Many are beneficial and symbiotic, and they're found in the human gut. So there's a lot here. Let's go through these. We have E. coli. Okay, that's in the human gut. It helps with digestion, but some strains cause disease. Okay, so not all E. coli are beneficial. Some are good, some are bad. Salmonella, that's most likely found in food poisoning or typhoid fever. Okay, we'll talk more about the bubonic plague. I think it's maybe next week. Lung infections, cholera, sounds like a lot of stuff. And then you have all this, there's the chromatium, and that's a sulfur-producing bacteria that oxidizes sulfur, producing H2S. Rotten egg smell, right? So there's bacteria, in hot springs and things like this that produce like this rotten egg odor. Okay, this would be it. Gamma Proteobacteria. Now, some of these form spores. So Mycobacteria, okay, generate spores. This next one is a sulfate-reducing bacterium. Okay, so they look like that. Epsilonproteobacteria. Many of these are in the intestinal tract of different animals. Excuse me. They've been found sometimes in deep sea hydrothermal vents or in cold seep habitats. Okay, maybe in the tundra or something like that. Okay. So, Campylobacter causes blood poisoning and intestinal inflammation. Not fun. Heliobacter pylori causes stomach ulcers. The biologist that discovered this had to convince his fellow colleagues that this bacteria can live in your stomach and irritate your stomach so much that it produces ulcers. Everybody did not believe him because how can bacteria live in a very acidic environment like the stomach? Now, the stomach is a very complicated organ with different layers and so on and so forth. But basically what he did was he cultured this bacteria, had it in a beaker, and in front of his colleagues, he drank it to their amazement. And then later on, he developed ulcers. And he had them examine him, and he proved without a doubt that this is the bacteria that causes ulcers. When you have ulcer treatment, sometimes they give you antibiotics to kill that type of bacteria so that your stomach will return to normal. There are a total of 52 types of bacteria. I don't want to spend the whole amazing day here going over everything, but let's just look at some other ones that are kind of bad. Chlamydias. All members of this group are intracellular parasites. They go into cells, take over the cell, and they make it their dwelling place, and it causes disease. Chlamydia trachomatis, commonly sexually transmitted disease that can lead to blindness if left untreated. And here's a zoom in of a pap smear. Basically, the pink color there are the included the cells that have gone inside, the bacteria that has gone inside to a cell. Okay, so that's a sign of a chlamydia infection. Spirochetes. I don't know how to pronounce these things, but most members are spiral shaped, okay? And some are pathogenic. So for example, we've got syphilis, the agent that causes syphilis, another sexually transmitted disease. Lyme disease. You may, if you're here in Virginia where I'm making this video, or maybe North Carolina or on the East Coast, you know we have a lot of deer, and sometimes ticks will latch onto the deer, pick up a disease called Lyme disease, and pass that along to humans. Now, they're not picking up a disease and passing a disease onto humans. They're actually picking up Borrelia burgdorferi, whatever how you pronounce that, okay? So it's in the blood of the deer, and the tick is a blood-sucking, insect. Well, it's an arachnid technically, but it's a bug, okay? It will suck the blood from the deer, and then it will have bacteria in it. And then if it gets on your dog, or if it gets on you, it's possible for the tick to bite you then, and then as it's trying to suck more blood from you, it transfers this bacteria into your skin, okay? And that's called Lyme disease. Cyanobacteria, like I said, is also known as blue-green algae. And these bacteria are similar to plants in that they are photosynthetic. So when you shine light on them, they develop their own energy, okay? And it's believed, now let's look at this. Prochlorococcus is believed to be the most abundant photosynthetic organism on the planet Earth, and it's responsible for generating half the world's oxygen. That's worth repeating. This little cyanobacteria guy here is responsible for generating half of the world's oxygen. Now, I'm not talking about in the past, I'm talking about yearly production. Now, when you hear about things like the rainforests being cut down or burned or transformed into farmland, you get concerned because, plants, all this green vegetation is supposed to be through photosynthesis, producing a lot of oxygen for us humans to breathe. But actually, most of our oxygen is produced by cyanobacteria living out in the oceans, which cover 3/4 of the planet Earth. So pretty interesting there. Gram-positive bacteria is a type of bacteria that has a certain type of cell wall, and so it's positive when treated with this stuff called gram stain. All right, so it's got a pretty tough cell wall, so it's sometimes hard to kill. So we've got Bacillus anthracis, which is anthrax-causing bacteria. We have botulism, which is, again, a food poisoning kind of thing. This Clostridium difficile, it causes diarrhea during antibiotic therapy. And we'll talk more about antibiotics, but when you treat yourself with antibiotics, it kills all the bacteria. And then maybe you're like, oh, I feel better. I'm not going to finish the bottle like the doctor said. Well, guess what? Those resistant bacteria, or some bacteria that can survive that, will then cause you problems because it's not your normal body intestinal flora and fauna, that's in there. And we'll talk more about this later, but Clostridium difficile, maybe it's difficult to kill that bacteria with antibiotics. It stays around after the therapy, and it causes you stomach problems, okay? Streptomyces. We've all heard about strep throat, and this is the guy here that does that. Mycoplasmas, these tiny bacteria are very small, okay? And some are pathogenic. All right, so what about archaea? Okay, so remember, Ikea and archaea rhyme. So we remembered again, we have those different domains. We've got the bacteria, which we've discussed on the bottom, and now we have the archaea. And you can see there's 4 different types, and there's a table here. We'll get into each of these. Now, the thing to remember is that all archaea tend to be extremophiles. And so we're going to learn about some crazy stuff here. First off, we've got, I cannot pronounce these words very well. Archaea, okay? So, Methanogens. Okay, methanogens cause flatulence in humans and other animals. So if you're passing some gas, you can blame your methanogens. Halobacteria. We talked a little bit about this guy here that was in the Dead Sea, I believe. It's reddish, slightly reddish due to the bacterial rhodopsin that is present in it. It's basically a certain protein that's red colored. Anyways, it's a red pigment, and that lives in salty environments. Here's another one. I won't try to pronounce this one. They play an important role in the fixation of carbon. Okay, some of these are sulfur-dependent extremophiles. Some are thermophilic or hyperthermophilic. So what do we have? Sulfolubus. Members of this genus grow in volcanic springs at temperatures of 75 to 80 degrees Celsius. What is that in Fahrenheit? Well, my hot water heater gets up to 45 degrees Celsius. I think it's 40 or 45 degrees Celsius. So 80 degrees is like twice as hot as my hot water heater. So it's not quite boiling, but it's pretty scalding, okay? So here's a archaea here on the right, and there's these little stick things, these little white features coming off the side of this are a bacteriophage. So it's interesting that a bacteriophage can infect an archaea. Anyways, I just saw that and thought that was interesting. Okay, by the way, we will talk about bacteriophages and viruses in the next week's class. Nano Archaea. Okay, so this group contains only one species. It was just such a weird thing that we had to put it in a whole class by itself. You know how sometimes you've been in a really small class, there just aren't a whole lot of students in that class. Imagine you're taking a welding class or a business class, and like you're the only student in there. Well, it would still be a class, and you wouldn't know what the typical student looked like because there's just one person in there. It's you. This is it. Okay. It's isolated from the bottom of the Atlantic Ocean from a hydrothermal vent, and it needs a symbiotic host cell to survive with, so extremely difficult to grow. Next up, we've got the core Archaea. Members of this phylum are found in only one place, something called the Obsidian Pool in the hot spring of Yellowstone National Park. There's some water there, there's some steam coming off that, and I guess it's called the Obsidian Pool because it's black like obsidian, or maybe it has obsidian in there. I didn't read up more on this, but they couldn't culture this, but they could pull some stuff in, throw it on a microscope and look at it and analyze it. And this is a micrograph of that, okay? So that in a nutshell is a little bit about what bacteria, what archaea is or are, what they do, where they're found. In the next lecture, we're gonna discuss a whole lot more about some of the minor differences of these two, a little bit about how they reproduce, their cell wall, stuff like that, okay? Thanks for watching. If you found this video at least a little helpful, please like so others will know that it's a useful video. Give a thumbs up, like, and subscribe. Thanks a lot, and have a great day.